Good evening, everybody. Thank you very much for your patience tonight. My name is Tom Ulrich, and I'm the Associate Director for Science Communications here at the Broad Institute, and it is my pleasure to welcome you all to this evening's Broad Discovery Series event. If you're joining us for the first time, the Broad Discovery Series explores the genetic and biological roots of human health and disease, the mechanisms that govern how our cells and our bodies function, new technologies that are changing what's possible in science, and progress being made to translate new discoveries into treatments for common and rare diseases. Tonight, I am excited to be introducing Miriam Udler and Melina Claussnitzer. Miriam is an associate member of the Broad, a clinician at Massachusetts General Hospital, founding director of the MGH Diabetes Genetics Clinic, and an associate professor at Harvard Medical School. Melina is an institute member of the Broad, co-director of the Institute's Type 2 Diabetes Systems Genomics Initiative, Associate Director for Scientific Strategy of the Novo Nordisk Foundation Center for the Genomic Mechanisms of Disease at the Broad Institute, and an Associate Professor at Harvard Medical School as well. So Miriam and Melina's research centers on three overarching questions. Why is type 2 diabetes different in different people? What can genetics teach us about that? And how can we use new genetic discoveries to better understand the biology of diabetes and create new treatment options for doctors and patients to use in the future? Before we begin, I want to take a moment to pause and acknowledge that tonight is also the 2023 Eliana Hechter Memorial Lecture. The lecture is delivered in honor and memory of Eliana, who is a gifted MD PhD student and member of the Broad community. And the lecture is supported by the Eliana Hechter Memorial Fund. If you have any questions for Miriam or Melina, flag one of our mic runners during the Q&A portion of the evening, and they'll come to you with a microphone so you can answer your question. If you're watching online, I'll be keeping an eye on YouTube chat, so please ask your questions there, and I'll do my best to work them into the conversation as the night goes on. If you're a Brody or if you're a scientist with a deep technical question for our speakers tonight, we ask that you please hold that question until the reception and approach our speakers then so that members of the local community have an opportunity to ask their questions and have them answered. Speaking of the reception, I invite you all who are here in the room to join us for a reception in the Broad Discovery Center right outside the auditorium here after tonight's talk is done, and I encourage you to explore the center's exhibits and interactive elements. That all being said, Miriam, please come up.
Great. Hi, everyone. My name's Miriam Udler. Thank you so much to Tom for the introduction and to the organizers. We're so thrilled you're here with us. Thank you all for braving the weather. For the people here in person and those of you joining online, welcome. Thank you for joining. So actually, before I start, I just want to take one moment also just to send, on behalf of Melina and myself, our appreciation to Eliana's family and loved ones for having this lecture in her honor. We've read about her and what a remarkable woman she was, and we're truly inspired by her, and it is an honor to be part of this lecture tonight. So I'm a diabetes doctor at Massachusetts General Hospital, and I see a lot of patients with diabetes, and it's a very common condition in the United States. Approximately 1 in 10 people have this condition, and so therefore, chances are either you or a loved one likely has this condition. And across the globe, we recognize that this is an epidemic. There are hundreds of millions of people affected, and the numbers keep rising. So why do so many people have type 2 diabetes? And you may have thoughts in your mind. I can tell you a few years back, I was at a conference, a research conference on diabetes for clinicians and researchers, and I was walking down a street heading towards the convention center in New Orleans, and I was thinking about diabetes, and this is what I saw. It's sugar. So a lot of people think that's all that is the reason for why people have type 2 diabetes. And in fact, it's much more complex. And if it were that simple, I don't think we would be spending our careers studying this. And so that's part of what we'll be talking about tonight is more of this complexity. So diabetes, type 2 diabetes is a condition of elevated blood sugar or blood glucose. And we tend to think of two primary reasons why the blood sugar will get elevated. One is that the pancreas in the body, the organ that makes insulin, is not making enough. And so insulin is the hormone that regulates blood glucose, and if you don't have enough insulin, the blood sugars will rise. At the same time, there's also a contribution that oftentimes people will have a lot of insulin circulating in their blood, but the tissues aren't responding properly. So there are a number of tissues that are meant to respond to insulin and pull in sugar, and if they're not doing that, then you have elevated blood glucose and insulin, and this is considered insulin resistance. And when blood sugars are elevated for extended periods of time, what we really worry about are the complications of diabetes, and these are really devastating complications. So type 2 diabetes is a leading cause of stroke, heart attack, kidney failure and dialysis, blindness, and limb amputation. So with such a common condition and such extensive, debilitating complications of the disease, it may surprise you to hear that we actually don't have a diagnostic test that can specifically say someone with high blood sugars has type 2 diabetes. And what happens in practice is that it's essentially a diagnosis of exclusion. So I'm a diabetes doctor. If somebody comes to me with high blood sugar, I'll be thinking, is there another reason to explain why their blood sugar is elevated? Do they have autoimmune diabetes or a medication causing their blood sugar to be elevated? And if not, then everybody is lumped together into this category of type 2 diabetes. And this is the vast majority of adults with diabetes, over 90%. And so sometimes we think of this as a waste bucket diagnosis. And then as a result of this, as you can imagine, there's a lot of variation across individuals with type 2 diabetes. And we call this heterogeneity, that there's differences in terms of how they present to the doctor, their course of their disease, and how they respond to treatment. And there are a large number of factors at play that contribute to why a person develops diabetes. So as I mentioned, it's not just sugar in the diet, it's not just obesity. There are a number of genetic components that are both protective and increase risk, and then also a number of environmental factors. And we're increasingly recognizing the importance of social determinants of health that impact whether people develop the disease and also their disease course. So I can tell you when I see patients in clinic, there are still a lot of fundamental questions that are unanswered. And these include, why is it that that patient in front of me has developed type 2 diabetes? And again, it's not just obesity. Although I do want to say that we often think of obesity as a result of people having an unhealthy lifestyle or unhealthy diet. And we're also increasingly recognizing that obesity is a disease in itself and that there's many contributions, neurological contributions. But again, type 2 diabetes is not a disease that just occurs because people are overweight or obese. And then I also ask myself when I see a given person, is there a subtype of type 2 diabetes that I think this person might belong to? But as yet, we don't have defined subtypes of type 2 diabetes. And then what treatment is best for that given individual? Now, there are very well-developed guidelines for how to treat a person with diabetes that have been developed by societies, experts in the field in Europe and the United States that have come together to create guidelines. And I'm showing these guidelines in the background here and purposely grayed out because the details don't matter. What you should know is that they actually don't currently take into account in any way the underlying cause of why the person has type 2 diabetes. And furthermore, the real focus of these guidelines is about lowering blood sugar. Again, but not recognizing or considering why the blood sugar is elevated, but just focusing on the end manifestation of the disease, which is the blood sugar. So the way that we often think about this is that because we're not looking at the underlying cause of the disease, we're essentially dealing with a situation where our management is suboptimal because we recognize there's a problem, there's a leak in the ceiling, the water's coming down, but we're perpetually just putting a patch over that leak and we're plastering it over, but the water's continuing to come down, the underlying cause is not being addressed, and so we're not really fixing the problem. So our vision for precision medicine in type 2 diabetes is that we could do better, that we can start to recognize subgroups of people where there is some underlying cellular process driving their disease. And then if we can understand that, we can start to target that for treatment that can improve our management of people with diabetes. So the idea is that rather than just perpetually putting a patch or plastering over that leak, we can start to think about the pipes above the ceiling and the reason why that leak occurred and try to hone in on the source of the leak. Now, this is challenging because, as you can imagine, we don't know the pipe structure above the ceiling. And the pipe structure is likely very complex. So this is where genetics can help us. So I'm going to take a moment just to give you a little bit of an overview of how we think about genetics of diabetes. So there's been a tremendous amount of discovery, and the discovery goes along a range of different types of genetic variants. So there are variants or mutations that are quite rare, that might only be seen in one family. And then there's also genetic mutations that are very common and that all of us will have. And the idea is that the variants that are very rare, these genetic mutations, may be seen in one family but have a very big impact. And if a person in that family has that mutation, they're very likely to have diabetes. On the other hand, these common variants have a very small impact, but they're seen in many, many more people. So they're potentially more relevant to the general population. So for a moment, I want to zoom in on this one example of a rare form of diabetes to help illustrate how genetics can help us understand disease mechanism. So what I want to tell you about is a very rare mutation in the gene PPAR gamma. And what happens is we know that PPAR gamma is a gene that acts in fat cells or adipose cells. And when this gene is faulty, we have impaired fat cell development. And with impaired fat cell development, we see development of, we see that people develop diabetes or lipodystrophy. And so this impaired fat cell development is the underlying disease process that's disrupted that helps define a subtype, albeit a very rare subtype, of diabetes. And what I'm showing you here is an example of someone with diabetes or lipodystrophy caused by this very rare variant, PPAR gamma. And what the clinical features that we see in these individuals are that they have reduced body fat, and particularly reduced body fat under the skin, or subcutaneous body fat, reduced good cholesterol, HDL cholesterol, elevated triglycerides, and insulin resistance, but without the necessity of obesity. So the idea is that those particular mutations that cause this rare form of diabetes are an example of being able to get to the source of the leak in the pipe. And if we go through our paradigm here, our vision for precision medicine, we can, this is an example of a, albeit rare, subgroup within diabetes where we recognize the cellular process or the disease mechanism is impaired fat cell development. And that can lead us to a treatment. So in the case of PPAR gamma induced lipodystrophy, and there's now drugs that have been developed that actually target the gene PPAR gamma. And this class of drugs are actually a very well-known class of drugs called TZDs. This particular drug is called pioglitazone. It's FDA approved. That not only treats the rare lipodystrophy, but it's actually a drug available to treat type 2 diabetes. So our hope is that through this process, we'd potentially find drugs that may be relevant to a particular subgroup and may also be relevant to a large proportion of people with diabetes, recognizing that many people will have disrupted pathways or processes that are more than one or multiple. So then going back to this schema for genetic contribution to diabetes, there's been, so as I mentioned, there's been a lot of discovery, and the most discovery for diabetes has actually come in this space of the common variants. So these are genetic variants or changes that are seen in more than 5% of the population. The way that these are discovered are through studies called genetic association studies. You may have heard the term genome-wide association study, or GWAS. So these studies take thousands of people, and they involve looking at DNA changes across the genome in all these people. And the actual analysis is very simple. All they're asking in these analyses is, for a given genetic change, is it different in frequency between people who have a disease and people who don't have a disease? And through this approach, there's now been over 500 different genetic regions associated with type 2 diabetes. This plot is showing over the years, over the past 2 decades, that there's been a number, these are showing the different publications, and that you can see the circles get bigger. The circles represent the size of the studies. Oops. And the circles are getting bigger. They're going from thousands of individuals to most recently over 1 million individuals as part of these studies. And then also the circles are changing in terms of the color, where you can see that purple represents people of European genetic ancestry. And over time, these studies are becoming more diverse, although still it's an issue that is very well recognized in the field. It's a problem that the studies are still biased towards people of European genetic ancestry, and there's a real effort to try to improve diversity. So what can we do with all this discovery? Over 500 genetic regions associated with type 2 diabetes. Well, there's a lot of interest in combining these variants together to generate a score to say what's someone's overall genetic risk for developing type 2 diabetes. And the idea, so this is a term called a polygenic score or a genetic risk score. So you can find people that have very few variants and those that have a lot of the genetic variants. And we know now that people that have the most number of genetic variants, the top 5%, have about a 5-fold increased risk of type 2 diabetes. And there's a lot of work to try to improve these scores and have it be a clinically useful tool for identifying people with diabetes risk. But what it doesn't tell us is very much about the person in front of us in clinic, or your family member or yourself who has type 2 diabetes, what kind of type 2 diabetes you have, because it's a mix of all these different disease processes all together. So we are interested in trying to take all those genetic variants and separate them out into the relevant disease processes, or the pipes above the ceiling. So if we can start to put the different genetic variants to make up the different pipes, and we can start to understand what the pipes are. The problem is this is not easy, and the reason that this is very difficult is that it's unclear which genetic variants or genes are actually disease-causing. So my lab has worked on trying to develop an approach to be able to connect a genetic variant to the physiological function or disease process without necessarily having the knowledge up front of the gene or the cell type that's relevant. So our solution is that we can group variants together, and the way that we can do this is by looking at how all the different variants affect many other clinical factors. So I'll show you what I mean by this. So we can take a whole list of diabetes genetic risk changes and then see how those same risk changes affect a number of other clinical traits. How do they affect body mass index? How do they affect fasting insulin or lipid levels? And then we can group them together using machine learning or cluster analysis to get groups of the variants that are most similar to each other in terms of how they're associated with all these different clinical parameters. And then by looking at those variants and the traits that define the groups, so for example, some of the variants may be associated with increased fasting insulin and increased body mass index, we can see the pattern and infer what the mechanism is likely underlying it. So using this approach, we have identified 5 salient genetic processes relevant for type 2 diabetes. So we published this work about 5 years ago now. And one thing that was very reassuring was around the same time, another group completely independent from our group performed an analysis and identified the same 5 processes. So these processes relate to forms of insulin deficiency. So there are 2 forms of insulin deficiency and 3 forms of insulin resistance. More recently, we've expanded this work to include hundreds of genetic variants. So we had started with 94. We published on over 300, and now most recently we have a manuscript with over 600. So the idea is that we're continuing to fill in with more and more variants to be able to complete the picture of the pipes and also identify additional clusters or pipes. So I'm going to, for a moment, focus in on one particular subgroup, which we've termed lipodystrophy subgroup. And the reason that we've termed, as you'll hear, is that this subgroup is reminiscent of the rare lipodystrophy subgroup that I had mentioned to you before. So this subgroup involves depending on our iteration of our work, at least 20 genetic changes or genetic variants that we can make a score for just those genetic variants. And by doing this, we can start to use the genetics to stratify people with diabetes and see which people have the highest risk just for that particular genetic process. So by looking at the people that have the highest risk for this lipodystrophy group, we can then say, without knowing anything about them, we can stratify that group, but then say, okay, now that we have this group, how do they differ compared to everybody else with type 2 diabetes? And what we see is that this group of people in aggregate have significant clinical differences compared to other people with diabetes. They have reduced amounts of body fat under the skin. They have reduced HDL cholesterol. They have insulin resistance without being as overweight or obese as other people with diabetes. They have elevated triglycerides, high blood pressure, fat disease, and fatty liver. And so again, these features were reminiscent of the features that we saw with the rarer form of subgroup of lipodystrophy. And that's why we've termed this type 2 diabetes more common subgroup as a lipodystrophy subgroup. And the idea is that we can use this subgroup and other ones that we've now been able to define to start to move towards understanding the underlying cellular process that Melina will tell you about, with the goal that we can, as we understand these cellular processes, start to target them for new treatments. So again, our goal is that by identifying these different subgroups, we're going to be able to get to the actual pipes and start to really fix the problem rather than just patching over the leak. Great. Thanks.
Thank you, Miriam. Can you hear me? Does it work? Yeah, great. So a tremendous honor and pleasure to be here and present jointly with Miriam, dear collaborator and friend, and our teams are very closely working together to start understanding, redefining better type 2 diabetes subphenotypes, identifying the underlying cellular biological processes that are the starting point for us to think about tailored therapeutic interventions and treatment strategies that are better tailored to individual subphenotypes that we observe in patients. So I want to start out and just give you a notion of the tremendous opportunity that we currently have at our fingertips coming from genetic discoveries. You've heard Miriam talking about the spectrum of different genetic variation in the human genome that links to type 2 diabetes. And we do know that genetic discoveries are incredibly meaningful at informing and accelerating drug discovery. And let me just walk you through this figure here, which is not quite intuitive to understand. What you're seeing, and I want you to focus on this kind of red box of common diseases. We are focusing on type 2 diabetes as a common disease. And what you see is the relative amount of drugs that is currently in the clinic, that is applied in clinical care and is used in patients. And the kind of color coding between blue and green indicates the drugs that are supported by genetic evidence, and the ones with gray indicate the drugs that are not supported by genetic evidence. So the bottom line of this figure here is a summary that current drugs in the clinic, more than 2/3 of those are supported by genetics, right? Which is a big statement to make. And even more so, much more clearly, I think, is the fact that any drug that is based on genetic discoveries is 2 to 8-fold more likely to successfully reach patients and be applied in the clinic. So genetics is tremendously helpful at guiding us in terms of drug development. Now, with what this figure or this slide here is supposed to show is that the genetic variants that Miriam has identified and clustered into these diabetes subtypes are really the very foundation for us to start thinking about and identifying a comprehensive list of targets and mechanisms of actions that serve for this, guidance of drug development, right? Provided that we can actually understand what are the fundamental issues of these pipes leaking, these pipes being broken. That's not trivial. And the reason this is not trivial is that we are looking these genetic clusters that Miriam introduced, genetic variants that associate with the diabetes subtypes, their associations. And we do know and we appreciate more and more that association does not equal causality. And one way to look at this is when you look at this beautiful association, we will all agree we see a fantastic association here. When I tell you, and you can read it, what the x and y axis say, we will all agree that the number or the amount of chocolate consumption in a given country is likely not the causal driving factor making up the number of Nobel laureates in this particular country, right? So this is a beautiful association, but it does not equal causality. And this is the very, very bottom of my lab's research that we are addressing when we are embarking on translating these genetic variants to their underlying causal functional impacts, their biological effects that are then actionable in a way that we can start thinking about drug development. So the mechanism here, that is what we are about. And the mechanism that basically informs the targets, the proteins, the cellular, the biological impact that these genetic variants have. But it is hard. It is very hard to make this connection between genetic variants and their cellular functions. And the key issue is the interpretation. We lack the understanding of how these disease-associated variants alter cellular and physiological processes and by which genes they actually influence to do. So in our work, as I mentioned, the variant to function work that we are embarking on, we are trying to tackle exactly that. So what is the problem? Why is it so challenging? Why is it so hard to translate these genetic variants to their underlying cellular functions? When the big challenge is, kind of encapsulated by the complexity, the beautiful complexity of biology. You see here a highly simplified version of a cell, and still it is, indicating the kind of complexity we are dealing with. We see an orchestrated cellular network. We see a flow of information of receptors getting certain signals and being translated at the DNA level, at the gene expression level, and so forth, that eventually will have an impact on a cellular phenotype. So the way we sketch this is this kind of network in the cell, which has some kind of logic to it, because based on the dogma of biology that all of us know, there's an information flow that follows logic. In the cell, biology is complex but principled. And we know that genetic variants have an impact at the DNA level. They will modulate some level of gene expression in most of the cases, have an impact on protein levels, and will eventually impact a given cellular phenotype that relates to the disease progression and disease manifestation, right? So by following exactly that kind of logic, we have introduced in 2015 the so-called variant to function framework, where we are defining a 5-step model for connecting these genetic variants to their biological effects. And we think what's important is to establish causal relationships between the genetic variants that act and their underlying, the regulatory elements at the DNA level that they impact, the gene expression levels that are modulated, the cell types and cell states in which they act, and most importantly for the work that we are embarking on here, the downstream cellular functions that are modulated in the context of these genetic variants that make up these type 2 diabetes disease clusters. Now, we have not only introduced this variant to function framework, but of course we applied it. We started applying it on initially on the strongest genetic association with obesity, and identified 2 target genes, IRK3 and IRX5, and their underlying cellular biological processes that are affected in both fat cells as well as brain cells. And we have applied it additionally to a series of other different genetic risk loci in the context of type 2 diabetes and obesity, and more recently in collaboration with Miriam as well on 3 different genetic risk loci that are falling into this lipodystrophy process-specific cluster that Miriam talked about. What I'm going to tell you now is everything I'm telling you now is around this kind of lipodystrophy process-specific cluster and how we are biologically characterizing it in order to make it actionable. First of all, we have the V2F framework, the variant to function framework. We show that this blueprint from going from these variants to genes to cells to biological pathways appears to generalize. And that's not only us, that is many in the community who have shown that. But we also acknowledge that making these connections for each individual genetic risk locus is incredibly time-consuming. It's a hand-to-hand combat type of work. It typically takes 5 to 7 years to make all of these causal, or establish all of these causal relationships at the single locus level. So we have been very interested on thinking about strategies to accelerate and really harness globally the systematic, all of the genetic variants in the human genome that fall into these different diabetes clusters, and turn them into biological process and understand what is the underlying biology. And that's what I'm going to talk about now. So the connection of the global signal of variants using the polygenic risk scores that Miriam introduced and linking them to disease biology. The way we do this is that we are working with patient samples at a population scale, and I will come to that in a moment. For now, just bear with me and keep in mind we are working with cell samples that come out of patients, out of hundreds of different individuals that reflect these different diabetes subgroups. And then we use certain technologies to read out the gene expression levels, the levels at if the chromatin accessibility is changed at the DNA level, and importantly for this talk here, the cellular phenotypes, the morphological and cellular effects that we observe in these patient-derived samples to start mapping the disease cellular processes for these subphenotypes, with the hope that we can pivot away from treating high blood glucose levels as an endpoint to really a directed, targeted therapy of these cellular pathways that make up these genetic clusters. Now, the prerequisite for such an effort was to build a biobank, and that has been a long effort up to now and is beautifully blooming over at MGH. The bottom line is that we are building a population-scale biobank that we call CellGen Bank in collaboration between the Technical University of Munich in Germany and the Mass General Brigham. The bottom line of this is that patients undergoing surgery, we are receiving fat tissue from these patients. We are receiving 2 different types of fat tissues. The abdominal fat tissue that is surrounding the liver and associated with adverse cardiometabolic risk outcomes, and the fat tissue below the skin that Miriam talked about, where we know that a depletion of the fat tissue below the skin links to adverse cardiometabolic outcomes, particularly for subphenotypes of type 2 diabetes like lipodystrophy. So we are receiving these fat samples, and we have developed a highly standardized way of isolating mesenchymal stem cells out of these fat tissues that are then banked here at the Broad Institute, and then we can take in culture and start differentiating these cells so we have mature, differentiated, developed version of these fat cells from the skin as well as from the abdomen around the liver. In parallel, these patients are enrolled in the MGB Partners Biobank, where we are using blood for for genotyping, that we get their genome, their genotype information, and we have deep clinical data from these patients as well. So with that in mind, we have an opportunity at hand that lets us understand the connection between these polygenic risk scores and disease biology by basically taking cells that represent high polygenic risk scores as well as low polygenic risk scores from our biobank. And then we use new machine learning-based technology to find all the ways how these cells differ under a microscope, including the ways that we can see, but a computer can't see, right? So that will help us reveal the biology behind these polygenic risk scores. 2 examples here of 2 different patients that you see. There are clear differences. We are extracting this disease biology from from microscopic images through machine learning-based technologies to understand the biology behind these polygenic risk scores. Now, to enable that, we developed an image-based profiling tool, which has these 2 compartments, an experimental part and a computational pipeline. The experimental pipeline basically means that we are using 6 different dyes that are multiplexed in 4 channels in the microscope, and that allow us to not only inform morphological processes, but also and particularly so metabolic disease-oriented processes. After imaging, we are then using a computational pipeline that is modified from the initial cell profiler pipeline, where you see here the ground truth, the raw image, and here the segmented files, segmented images based on the nucleus, lipid droplets, the mitochondria in red, and the actin cytoskeleton. Now, the outcome of this, after the computational extraction of features, is a high dimensional representation of cellular processes in these patient samples or in these cells. We're extracting 3,000 features out of them that inform a series of different processes that is relevant for metabolic disease. And each of these processes is supported by hundreds of different features. So it's a deep dive, high granular look into what is going on there at the biological level. Just one very simple example, which is the fat cell differentiation or the development of a fat cell, where we are starting with these patient samples at day 0 of differentiation and then send them down the differentiation line with this progressive accumulation of lipids that you can see here. And indeed, the lipocyte profiler pipeline extracts features such as the intensity of these lipids that we can track throughout the differentiation trajectory, right? There's lots more to talk about, but I want to shift back again to our overall paradigm to connect polygenic risk scores to disease biology, and primarily so start mapping the cellular heterogeneity of type 2 diabetes that Miriam talked about. And that, I will guide you through 3 different scales of heterogeneity of type 2 diabetes and what this image-based profiling pipeline lipocyte profiler tells us about the biology behind the polygenic risk scores. The first one is type 2, the full type 2 diabetes polygenic risk score distribution. Miriam mentioned it's a highly heterogeneous trait. I'm using that basically to show you how we can improve from this. Insulin-resistant polygenic risk score, where we have the contributing organs, the liver, the skeletal muscle, and the fat. And then I will switch to our most favorite polygenic risk score, and that is the lipodystrophy process-specific risk score that Miriam and her team have identified, and we are using here. This signifies severe insulin resistance in the absence of obesity, rapid progression to coronary artery disease and chronic kidney disease, and so forth, so clinically highly relevant. Now, the outcome of connecting these 3 different polygenic risk scores that reflect the heterogeneity of type 2 diabetes through microscopic imaging and a feature extraction pipeline with lipocyte profiler gives us this result, and we can start seeing relationships between the scores and the actual cellular programs that we observe in the dish. So for type 2 diabetes, unexpectedly so, remember it's highly heterogeneous, we are getting almost no signal whatsoever. These spider plots basically represent the number of features that robustly, statistically significantly connects with these polygenic risk scores. So a lack of signal here for type 2 diabetes. We are getting a strong signal for insulin resistance in the lipid channel. So there appears to be a perturbation of the lipid metabolism here in visceral adipocytes. And when we turn to the lipodystrophy cluster, we preserve some of the insulin resistance signal in the lipid channel, but we are getting this very specific lipodystrophy signal that is primarily enriched in the mitochondria. The mitochondria, the powerhouse of the cell. Now, let me show you how the images look from patients from the bottom, from low polygenic risk for the lipodystrophy process-specific risk score, and patients with high polygenic risk for lipodystrophy. These are ground truth through images from patients, and they are computationally averaged. So we have averaged tens of thousands of individual cells from individuals on the left with a low polygenic risk for lipodystrophy and the right with a high polygenic risk of lipodystrophy. And it is astonishing to us. The right side, you see this very clear contrast in terms of the red coloring. The red color, the dye we are using here is mitotracker deep red, which informs and is directionally proportional to the mitochondrial membrane potential, which basically is the activity of these mitochondria in the skin fat cells. Now, these mitochondria, they appear to work too hard, and they are small. They're small and fragmented and dysfunctional, and as a result, most likely, these cells are not able to accumulate as much lipids, which is shown in green fluorescent color, as the low polygenic risk individuals. So there's a very strong contrast we are observing. Now let's look at the features themselves. There's a lot in this figure here, but I wanted to show you the signature of all the significantly associated features of the lipodystrophy polygenic risk score and the image-based profile. So what it comes down to, the higher up in the signature, the stronger the positive correlation or the relationship between these lipocyte profiler features and the lipodystrophy polygenic risk score that we are observing in patients. And when you look at the top of the signature, you see that the strongest signal is indeed related to the mitochondria. We are observing an increased mitochondrial activity, mitochondrial membrane potential, and an increased fragmentation of these mitochondria, as you can see here. So these mitochondria are very small and hyperactive, and we are observing a decreased lipid accumulation. So we can start with that now, placing these lipodystrophy polygenic risk scores on a path, on a sequential path, where the polygenic risk score appear to modulate gene expression level that is informative for mitochondrial dysfunction. I have not shown you data on the gene expression levels, so you have to just take my word for granted now. But we see that these propagate to features informative for mitochondrial hyperactivity phenotype and an elevated mitochondrial fragmentation linking to insulin resistance. So coming back to Miriam and my lab's vision for precision medicine in type 2 diabetes, we are now we are using these genetically defined disease subgroups from Miriam. I've shown you examples for the lipodystrophy subgroup of patients. We are identifying the genetically anchored cellular processes that underlie that with the hope to open a path towards developing new treatment options. And we have started embarking on that. When we look at this particular figure here, the point I want to make is that the tool that we have been using for learning about disease biology behind these lipodystrophy polygenic risk scores could be used also for identifying new treatment options. So this hypothesis we tested in a recent effort as part of the CPGEM consortium here at the Broad Institute, where the hypothesis is that if a compound makes cells, these subcutaneous, these skin-fed cells, through microscopic imaging, look less like the high polygenic risk individual samples and more like normal cells, then this might be a good starting point for us for a new drug that would specifically work for people with that particular form of type 2 diabetes, right? And indeed, we can start making these observations. I just show one particular example here of an inhibitor that's called TCS7004. This is an inhibitor of the gene DYRK1B. And when we are exposing the cells to this particular inhibitor, indeed the cells appear to look more like cells from low polygenic risk individuals and less like cells from high polygenic risk individuals, as a great new option or opportunity to think about treatment strategies. So coming back to this paradigm here of linking disease variants to biological functions and therapies, I hope what we have shown you today is by starting to cluster variants into type 2 diabetes subtypes that form these distinct groups of genetically anchored phenotypes of type 2 diabetes, and linking these phenotypes to their cellular processes opens the path towards developing new treatment options and better subtyping of type 2 diabetes patients. So with that, we wanted to thank particularly our labs. I think, should I say that on behalf of both of us? Tremendously grateful for the work, the hard work my lab is doing, and I know Miriam feels exactly the same way. We love to work together, and we hope to make, to help in the path of defining new treatment strategies for type 2 diabetes. Thank you so much for your attention, and I'm looking forward to questions.
You stay up here. You're not going anywhere. Coming up, Miriam. So this would be the time when you guys, if you have questions, please ask. Just raise your hand, and my colleagues Val and Namrata will run over to you with a microphone. And as you're gathering your thoughts and coming up with the insightful inquiries that you're going to be peppering our speakers with, I wanted to start off with a question for you, Miriam. So my family we have a history of type 2 diabetes. It runs in the family. I'm at high risk. Unfortunately, my children are probably also at high risk. The subtyping work that you're doing, the genetic discoveries that you're making, how far away are we from having that knowledge applied in the clinic to help people, well, basically people like me and others like me?
That's a great question. So we still have a little ways to go. The subgroups that I described exist. The way that we see that the subgroup is clinically different from other people is in a group level. So we still, within that subgroup, would not be able to recognize one person as different on their own compared to everybody else. And what's really remarkable, though, is that Melina's research is showing that the individual level, probably the cellular differences would be apparent, but at the whole body level, we just can't see the differences clinically. So we're not there yet. I think there's a lot of, it's a very important direction that we hope to go into, and you can imagine that maybe that would involve eventually developing diagnostic tools that could get to more close to the genetics as opposed to just the whole person's clinical features. But I think our hope is that we'll eventually move in that direction, and then but in parallel, still be moving towards the treatments, because even if our hypothesis is that all the treatments we're developing will be useful for many patients, so that even if we still get to the point where we can say, we might be able to say, for you, there's these different contributions. We might not be able to put you in exactly, or your family members into one bucket, but we might be able to say, okay, in your family, we notice that these different genetic scores are high, so you're likely to respond to these treatments, and it'll help us target the treatments.
Melina, question for you as well. So the framework that you developed and all the work that you've been doing here, it sounds like right now it's all at like one variant at a time kind of level? Or is there work to try to create mechanisms by which you or others could look at hundreds, dozens, thousands of variants at a single time and kind of accelerate this process of getting through that gigantic list of variants that's coming out of the genetic work that people like Miriam are accomplishing?
Yeah, that's a great question. So beside the aggregation of variants into polygenic risk score to kind of capture composite scores and all of the variants at once and link them to disease biology, we are also developing strategies, and not only us, but many here at the Broad and in the international community, to accelerate these efforts by increasing the scale of CRISPR perturbation screens, by running these kind of natural genetic variation studies, as we call them, that you have seen working on patient samples, scaling them by pooling these individuals into individual dishes, scaling these studies to many hundreds at once, and reading out with different phenotypes. And then certainly so an important component that we are embarking on more and more is the development of machine learning models that help us make predictions matched with the right data generation projects, massive data generation projects that enable a high hit rate of these machine learning models. Yeah.
We have a question from the audience. You, sir.
Hello. Can you hear me? Yeah.
Yes.
So type 2 is the end disease, and your work is on the genetic side of the house, what causes what. Does lifestyle play any role in this, or should I just wait for your research to produce drugs?
I'm sorry, I couldn't hear it. Does what play any role?
Lifestyle.
Oh, absolutely.
And what can one do about it?
Great. So, well, the simple answer is that everyone benefits from lifestyle interventions by having a healthy lifestyle. So even people without diabetes would benefit from that. And it's, there's so many health benefits in terms of lowering risk of heart disease and stroke. And so the easy answer is that everyone will benefit no matter what your risk for diabetes is. What we know is that certain people, it probably is more important than others, meaning that certain people may have a higher genetic risk for diabetes, and so if they're at a higher genetic risk, then you really want everything else you possibly can control to be in your favor to help prevent the development or to help treat the disease. So I guess there are two factors. One is the development of diabetes and having lifestyle interventions to help prevent that, which we know works very well. But then once you have the disease, lifestyle interventions to help control diabetes. And so I think no matter, I mean, the message really is that right now that is the benchmark. Like everybody should be trying to have as healthy lifestyle as possible, but that some people are, no matter how hard they work at lifestyle, they will still be at increased risk of diabetes. And that's where I think our work really comes in, is that I mean, I can tell you I see people all the time in clinic who say, I'm doing everything. I'm running, I'm eating really healthy. I don't understand, why do I still have diabetes? And it's because there's other factors that they can't control. And that's where hopefully we'll have, with understanding the genetics, we can have better treatments to offer.
Up in the back there.
Hi, thank you. Great talk. I'm wondering what is the reason or why the mitochondria are changing in this lipodystrophy. What are they fragmented? Why are they hyperactive? What is the mechanism going there?
That's an excellent question, and there's a lot of hard work going on in the lab right now to understand all of the contributing factors. We do know what it is not related to. There is a physiological process in adipocytes that's called the beiging project, or browning of adipocyte, which is the uncoupling of, or driven by uncoupling proteins. That is what we are not observing in these skin fat cells, an elevated activity of the mitochondria through an uncoupling phenotype. We don't know what brings the cells to, or the mitochondria to fragment, to no more fuse in these large tubules that are working properly at the normal level as we would expect it, but start a process called fission, where we have smaller fragments of these mitochondria that don't work well. We have some early indications that it's related to a process called senescence in these adipocytes, which is an age-related, or handled as an age-related phenotype. But I don't want to comment too much on it because we don't have enough data to back up that statement yet.
Melina, we had a question from one of our viewers on YouTube. It is, how are patients identified for participation in CellGen Bank? Do they need to have been diagnosed for a particular length of time to qualify, given that the type 2 diabetes, as you mentioned, is a diagnosis of exclusion? I, too, am quite curious about this.
That's a fantastic question. So we have CellGen Bank currently, as it is running at MGH at the very moment, enrolls patients through the Department of Surgery. So indeed, a patient has a particular intervention, and with that, we are receiving the tissue. But we have an IRB approval in development to start recalling patients and to start onboarding patients for skin biopsies to get these fat samples from below the skin from really anybody who is interested. That's not quite through yet, but we are hoping to get there very soon.
All right. And then another question that we had here was, with 3,000 features defining a point in your data set, have you considered alternatives to averaging? Are you doing any kind of other analysis on the averages? It seems like the secret is not being observed.
The secret. So averaging, I assume this is averaging features or collapsing features into individual processes. That's a great question. We have actually the person who is running the analysis here, Hissam Dashti. We are developing a series of different feature selection tools of the 3,005 features, which is a high-dimensional space. What we really are interested in are the driving features and offset them from any type of secondary features that kind of are just standing as a proxy for that process. There's a lot of mathematical thoughts going into this to develop these type of feature reduction or feature selection pipelines borrowed from the gene regulation realm of research and statistical genetics methods as well. So we hope we can answer more very soon.
I think we have a question down here.
Thank you for the great talks. My question probably is addressed towards Miriam. So both talks show us that sometimes we need very specific cells from specific tissues because genes might have specific function or dysfunction relevant for diabetes in them. And then linking back to your first initial question, like this unmet need in large cohort of diabetes type 2 patients, how would we translate this knowledge from this cell intensive workflow towards back to the patients. What's your best bet to the future? Like, should we perform biopsies to such patients and classify them somehow, or you think it's more about liquid biopsies and we should work in that direction?
That's a great question. I think certainly we can use the DNA signature. So we can one thing we can do is, without having to be invasive, we can, from just a blood sample, we can identify people that are high risk for any of these genetic subgroups. So that's, I think, a starting point. There's also, we're really interested in trying to understand various signatures that relate. So Melina has described really beautifully the cellular-based signatures, and there's expression signatures that would involve more invasive tissues. There's also signatures related to metabolites, which are, so the idea that there might be other biomarkers in the blood that might also help connect people to subgroups. So that's ongoing work as well.
Miriam, I had one more question. You mentioned early on in your talk about variant discovery and the differences in the variants that you were seeing across different populations of different genetic ancestries. Can you talk a little bit more about why that's important, how genetic ancestry fits into the picture that you're starting to see?
Sure. So the more diverse populations are, the more you're going to see different genetic variants that might be only seen in one population and not seen in another. So the more genetic diversity, and therefore population diversity, that is studied, the better we are to really capture all possible, maybe to use the analogy, all possible little metal bits constructing the pipes. So I think that's, and there's examples where there are really important genes. Like there's a really beautiful example from an Inuit population in Greenland, where there's a very important gene that is that people there have a mutation in, and this gene mutation you don't see in other populations. And so you would never be able to connect that gene in this genetic subtype to diabetes without having that population. So the more we can be inclusive and improve the diversity of our genetic studies, for sure that's going to help accelerate discovery.
And then I want to ask one last question. Melina, what is the biggest challenge that you see in making those connections, in putting together those links between genetics and biology, and in the end, disease symptoms? Like, what is the biggest hurdle that you think you have to jump over?
Oh, wow. That's the problem is there's so many challenges. Pinpointing one. I would say more the need for transdisciplinary efforts. So as part of the Novo Nordisk Foundation Center here at the Broad Institute, what we deeply think about is how can we define experimental setups that are machine learning aware and that will help us train the next generation of machine learning models that will help accelerate this effort of linking genetic variants to their underlying functions. So that requires some shared language and deep discussions between those excellent researchers and scientists developing experimental strategies and those computer scientists that develop machine learning models. So I wouldn't frame this as the biggest challenge, but I would frame it as at least one of the biggest requirements, I think, to really scale the extracting information from these genetic signals at a higher scale.
Well, I think it's time for us to step out and have some refreshments. So thank you, Melina. Thank you, Miriam. And thank you to all of you for coming out on this cold and raw night